Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So today we are going to discuss about the examples of imprinted genes. So in last class when we were discussing about genomic imprinting, we learned that there are certain genes which exhibit monoallelic expression which is dependent on their parent of origin, and those genes are known as imprinted genes. Means depending on from which parent that gene is inherited, may it be father or may it be mother, depending on that, those genes will be either expressed or silenced, and thus those genes are known as imprinted genes. Again, in last class, we discussed about parental conflict hypothesis. So, how both of the parents, mother and father, they have different opinions on how a comfortable growth should be for their progeny to reach at the certain reproductive age and continue their legacy, continue their future generation. So we learned in that class that the paternal copy of gene, it supports growth. So those will be growth activating genes, while the maternal copy of genes, they will suppress the growth. So that they will promote certain factors which suppress the growth. Okay. So that's all we learned in last classes. Today we will learn about the examples of imprinted genes such as the KCNQ1 locus, the H19 IGF2 cluster. So how those imprinted genes play a role in fetus development, that we are going to learn today. So for that purpose, let's first learn about imprint control region. So this is crucial. This is very crucial region. So imprint control region is abbreviated as ICR. Okay, so this is important because this imprint control region, as the name suggests, it controls genomic imprinting. Okay, so that is why we are discussing about what is imprint control region. So many imprinted genes, they are present in cluster, and that cluster forms the imprint control region. So for example, this is a chromosome. Okay, this on this chromosome, on this locus, there is a region present. Okay, and this is characterized by DMR, differentially methylated region, and around which there are multiple imprinted genes. Okay, so let's say this is gene imprinted gene 1, 2, 3, 4. All of those are imprinted genes. Okay, and all of those, they are controlled by this imprint control region. Okay, all of them, they are controlled by this imprint control region. So this cluster, this whole cluster of genes, imprinted genes as well as the differentially methylated region, this is known as ICR. Okay, so this is what we call as imprint control region, and that is going to control the genomic imprinting. So let's discuss what is DMR. So DMR is differentially methylated regions, means that this region, this locus, this particular region, this is differentially methylated in the both copies, the copy which is inherited from mother or father. Meaning that it will be either methylated in maternal copy of the genome, or it will be methylated on the paternal copy of genome. Okay, so let's assume that this differentially methylated region, that is only methylated in maternal copy of the region. So we will call that imprint control region, that ICR, as maternally imprinted region. Okay, so we will call that those genes, those imprinted genes, or that ICR is maternally imprinted. Okay, if the methylation is only on the maternal copy of the genome. If the DNA methylation is on the paternal copy of genome, we will call that as paternally imprinted region. Okay, I hope this is clear. So this is differentially methylated. So only one copy will be methylated, either the maternal copy or the paternal copy. And the copy which is methylated, we call that it is the imprinted copy. So maternally imprinted genes means the methylation, the ICR will be methylated, but only the one which is inherited from mother. So only maternal copy of genes, they will be methylated, and that is why they will be called as maternally imprinted or maternally imprinted ICR. Okay, maternally imprinted genes or maternally imprinted ICR. So how is this differential methylation is exhibited? So this differential methylation is done during the gametogenesis. Okay, I hope when we were discussing about the dynamic changes that occurred in the DNA methylation during development. I hope you remember that. The very first event that the embryo undergoes is the global demethylation. Okay, so what happens is that when the egg and sperm fuses, so when the egg, so this, let's say that this is the maternal, pink one is the maternal copy of the genome. Okay, so this is the maternal copy of genome, and this is the paternal copy of the genome, the blue colored one. Okay, so when both of them fuse together, so they form embryo, they form zygote. Okay, zygote which undergoes cleavage and form the embryo. At that point of time, both of them will exhibit their own DNA methylation patterns. Okay, because they are the gametes which are inherited from the parents, so the DNA methylation will be high. So on the y-axis, we are showing the percentage DNA methylation in the cells, okay? And on the x-axis, we are showing time, okay, the developmental time. So the DNA methylation will be high during fertilization because we are receiving the genome from mother and the father. So they will already have certain DNA methylation pattern. So then they will undergo the global demethylation event. So whole methylation will be erased, and then that will be reset in the cells, okay, in the somatic cells. But when then the certain cells known as primordial germ cells, they will go and undergo the gametogenesis, means they will be forming sperm or egg. Though all of the methylation marks will be erased and they will be reset according to the sex of the baby. So let's assume that the embryo which is now formed, it is going to be male in future. So when the cells, primordial germ cells, they will go undergo the gametogenesis, so they are going to form sperms. Okay, so they will undergo spermatogenesis, and that is why the methylation marks that will be reset, that will be according to the paternal genome. Why? Because this sperm, this is going to form a part of paternal genome in the future progeny. So this is going to be paternal genome for the future progeny, and that is why the methylation mark will be set according to that. Okay, and if the embryo is going to be female, the new methylation mark will be set according to the maternal genome. Okay, so that's what we learned in that class about the dynamic changes in DNA methylation. So what happens is that this global DNA methylation event is only for somatic cells, only for the normal cells with normal genes, but the imprinted genes, they do not undergo any demethylation event. Why? Because this imprinted region, this should be maintained in the future progeny as well. So for ensuring that this imprinted genes should, they will be maintained in the future progeny, the DNA demethylation is not done. The DNA methylation is maintained. So methylation is protected in the imprinted genes until they reach to the gametogenesis stage. Once they reach to the gametogenesis stage, they undergo demethylation. Okay, so they will undergo demethylation and then again remethylation. Okay, so what happens is that during the gametogenesis part, okay, so this is the gametogenesis part, and this shows the imprinted genes. Okay, methylation on the imprinted genes. So during gametogenesis, all the imprinted genes, they undergo demethylation, so that new methylation marks that will be set according to the gamete. So the new gamete, if it is going to be sperm, okay, all of the imprinted genes, they will be paternally imprinted. The pattern will be set according to the paternal genome. But if the gamete going to be formed is the egg, if they are going oogenesis, so the egg will have the maternal genome, right? In the future progeny, it is going to be a part of the maternal genome for the future progeny. And that is why the egg that will exhibit maternal imprinting. Okay, and this is ensured during the demethylation event. And that is why during the gametogenesis, those imprinted genes, they undergo demethylation, so that so that the imprinted genes, they will exhibit the imprinting, or they will exhibit the DNA methylation according to the gamete, according to the sperm or egg. Okay, so this DNA demethylation event, this ensures that differentially methylated regions, they will exhibit the DNA methylation according to the sex of the gamete. Okay, I mean the whether it is sperm or whether it is egg. So according to the sex of the individual. Okay, so if the individual is male, the gamete going to form a sperm, and that is why all of the differentially methylated regions, they will be paternally imprinted. Okay, so if it is paternally imprinted, it will be methylated, and if it shouldn't be methylated, it won't. I mean, let's get known about it in the examples. So let's learn about the KCNQ1 locus first, so that what I discussed will be clear to you all. So let's discuss about KCNQ1OT1. Okay, so let's discuss about KCNQ1 locus. Okay, so this is a very important imprint control region. Okay, and this is control. This cluster is regulated by a long non-coding RNA, LNC RNA, which we call as KCNQ1OT1. OT means the opposite transcript, the antisense transcript. So let me explain this cluster first. So let's say there is a genomic region on which this cluster is present. Okay, okay. So this is the KCNQ1 gene. What's happening? Let's say that this is the KCNQ1 gene. Okay, on this gene, there is a region, there is an intronic region. Okay, let's say this is the intronic region. Okay, and this is going, if this is transcribed, this is going to form KCNQ1OT1 long non-coding RNA. Okay, this region, this intronic region, this is going to form KCN1Q1OT1, sorry, KCNQ1OT1 long non-coding RNA. Okay, so this gene, KCNQ1 gene, this is present on both maternal copy and as well as paternal copy. Okay, as well as this OT1 gene also will be present, but this will be either methylated or unmethylated depending on whether that genome is part of maternal genome or paternal genome. So let me make 2 copies of this. Okay. So let's assume that the first one that we showed here. Okay. This genome, this is the part of the maternal genome. So in an embryo, this is the maternal copy of the genome, and this is the paternal copy of the genome. So this is the maternal allele and this is the paternal allele. Okay. So both of them will have KCNQ1 gene. Okay, which and this gene has KCNQ1OT1 long non-coding RNA. The intronic region which is going to code for this long non-coding RNA, we call that OT1 because it is opposite transcript. Means that is, this transcript is transcribed from the antisense strand of the DNA. Okay, so this is how the ICR, the cluster is present, and there are certain genes which it controls. Okay, out of which there is an important one that we will discuss today, and that is CDKN1C. So this is cyclin dependent kinase inhibitor. So this is inhibitor. CDKN1C. This is inhibitor of cyclin dependent kinase. Okay, so this is the ICR which is present in both maternal and paternal copy. So the KCNQ1 locus, this is maternally imprinted. Okay, so I told you before that if certain if it is maternally imprinted region, then the maternal differential methylated region will be methylated. Okay, so this is maternally imprinted gene. Okay, so the KCNQ1, KCNQ1 locus, this is maternally imprinted. So if it is maternally imprinted, so the DMR in the maternal allele that will be methylated. So the maternal allele DMR, and that DMR is KCNQ1OT1 here. In this case, the KCNQ1OT1, that is the DMR. So this DMR will be methylated. Okay. I'm showing this will be methylated and this will be unmethylated. Okay. So this representation, the shaded representation means it is the methylated locus, and if it is unshaded, it is unmethylated locus. Okay. So this is the unmethylated part. So this KCNQ1 OT1 long non-coding RNA, this is the DMR in this case, and this DMR is unmethylated in paternal copy and it is methylated in maternal copy. So what happens is that if it is methylated in maternal copy, means it is repressed. So if it is repressed, the KCNQ1 OT1 long non-coding RNA that will not be expressed. Okay. So in the maternal copy, this long non-coding RNA, because it is methylated, it will not be expressed, but it will be expressed in the paternal copy. Okay. In the paternal copy, the methylation is not there. So that is why the long non-coding RNA will be expressed. So what happens is that we learned in the long non-coding RNA series that the long non-coding RNA, they can act as a guide or they can act as a platform. So in the scaffold mechanism. So in both of the mechanisms, they recruit certain chromatin remodeling factors, or they can recruit certain enzymes which are going to change the chromatin environment. So they can either repress the genes, or they can either express or elevate the expression of certain gene depending on which chromatin remodeling complex they are going to recruit on the site. Okay, so this long non-coding RNA, this recruits enzymes known as G9A. And I hope you remember in the DNA demethylation, histone methylation series, which learned that the H9A, this is HKMT, means this is histone lysine methyl transferase. So this adds the methyl group on the lysine residue. Okay, so this does the H3K9 methylation, which is a repressive mark. This does the heterochromatinization as well. Okay. Again, this long non-coding RNA does the two jobs. This recruits G9A as well as it recruits PRC2 complex. I hope you remember that PRC2 complex is also this is also related to the silencing of gene, and it does the H3K27 methylation. Okay, so both of them are the hallmarks of or they are renowned for the heterochromatinization, means the repression. So both of them, they are going to repress certain gene that we discussed before, and that is CDKN1C. So they are going to repress CDKN1C. They are going to repress the inhibitor of cyclin dependent kinase. Okay, that means that when those G9A as well as PRC2, they will recruited by the KCNQ1OT1 long non-coding RNA on this imprinted region, the CDKN1C that will be repressed. So inhibitor of cyclin dependent kinase will be repressed. This means CDK, the expression of CDK will be elevated, or it will not be hindered by any inhibitor, means this will promote growth. Okay, so this is going to promote growth only if the paternal copy is active. So I hope you remember when we discussed about the parental conflict hypothesis, we discussed that the paternal copy of genes that is going to support growth, while the maternal copy of gene that is going to suppress growth. So how does maternal copy suppress growth here? So what happens is that the DMR, means the long non-coding RNA, that is methylated. So this is methylated. So though there will not be KCNQ1 or T1. So there will be no long non-coding RNA. So now there is no long non-coding RNA. So the repression of CDKN1C will not be there. Means the inhibitor of the cyclin-dependent kinase, which promotes growth. So cyclin-dependent kinase promotes growth. Means cyclin-dependent kinase inhibitor CDKN1C. This is going to suppress growth. So if there is no long non-coding RNA, so there will be expression of CDKN1C, and that is why the growth will be suppressed. So let's summarize it here. So what happens in the KCNQ1 locus? So let's see what happens in maternal allele and paternal allele. So the DMR, let's see what is the status of the DMR. So DMR will be methylated in the maternal allele, and that is why we call that this locus is maternally imprinted. Okay, and the DMR in the paternal copy, that will not be methylated, so this is unmethylated. Okay, and we know that the DMR here is the KCNQ1OT1. This is a long non-coding RNA, which is opposite transcript or the antisense transcript of the KCNQ1 gene intron. Okay, so that is a long gene, and one intronic region that codes for the long non-coding RNA that we call as KCNQ1 OT1, and that OT stands for the opposite strand, opposite transcript. So the antisense strand of DNA that is going to produce long non-coding RNA and this is the KCNQ1 gene. Okay, so the KCNQ1 OT1, this long non-coding RNA. Okay, let me just erase this. Yeah, so this long non-coding RNA that will not be produced in maternal copy, because the DMR which codes for this long non-coding RNA, it is methylated, so this will be absent, or let's say that this will not be produced in the maternal from the maternal allele. Well, the KCNQ1OT1 that will be produced by the, so this is present, or that will be produced by the paternal allele. Okay, so yeah, so CDKN1C, this is the inhibitor of the CDK, the cyclin dependent kinase inhibitor. So this will be present, or this will be produced by maternal allele. Why? Because there is no long non-coding RNA which is going to do the repression of this gene. So, okay, but in the paternal allele, this long non-coding RNA is it is present, it is expressed, and now this long non-coding RNA that is going to recruit the G9A as well as the PRC2 complex, which are going to again recruit DNMT, the DNA methyl transferase. Means all of them, they are going to recruit the repressive marks on the CDKN1C gene, and that is why this gene will be silenced. Okay, this will be silenced, mean that will, so this inhibitor will not be present. Okay, so if this inhibitor is not present, this means that there will be CDK, so that means the growth will be promoted. So growth will be promoted. Well, this is present, this inhibitor is present in from the maternal copy, from the maternal allele, and that is why growth will be suppressed. I guess I did something wrong. I don't know. I just can't remember the spelling. So the growth will be suppressed here. Okay. So I hope now you remember that when we discussed about the parental conflict hypothesis, how the growth suppression and how the growth suppression will be done by the maternal allele, and how growth promotion will be done by paternal allele. So growth will be suppressed. Okay, so we see the growth suppression in the maternal allele, while there will be promotion of growth in the paternal allele. So I hope this was clear to you all how the KCNQ1 locus, this imprinted gene, this KCNQ1 gene that regulates the fetus development. Okay, so this balance creates this balance is very crucial. If this balance is not maintained, there will be certain syndromes such as BWS syndrome, the Prader-Willi syndrome. We will discuss about that syndromes as well, but not in this lecture. So let's learn about other cluster, the H19-IGF2 cluster. This is a very important and renowned example of imprinted genes. Okay, let's learn about how this cluster, this is going to control the fetal development. Okay, so this cluster contains two genes, the H19 and IGF2. Okay, so this is the, let's say that this, okay, this is the region on which there will be a gene known as, which codes which is known as H19. Okay, let's say that this, so this is IGF2. We know that this is a growth factor. IGF2 is a growth factor. Okay, there will be other gene known as H19. Okay, and both of them, they are placed under the control of enhancers. So this enhancer, this is controlling H19 as well as IGF2. Okay, but here comes a twist. Between those two genes, there is a region here. This is known as CTCF binding site. So this is binding site for a protein which is CTCF. Okay, so this is a site which binds to a protein known as CTCF. And CTCF, this is a very important protein because this is an insulator protein. So this is insulator protein. So those proteins are the boundary elements. Okay, so they insulate two different regions located on the same chromosome. Okay, so this acts as a boundary element for both H19 and IGF2. So what happens is that, imagine this element as a wall. So when on the CTCF, so to this binding site, if the CTCF is bound, so let's assume that this is CTCF, and if CTCF is bound, it kind of creates a boundary here. So the enhancer can only control H19, but it cannot control IGF2 because it cannot cross this boundary now. So it's like a wall which is standing tall here, okay, standing between IGF2 and H19, and enhancer can now only control H19 because it has only control on the H19 because it can see H19, but it cannot see the IGF2 because of the wall here, so it cannot control the IGF2. So if the CTCF is binding here, it will only control H19. If CTCF is not binding here, it can, sorry, if the CTCF is binding here, it will only control H19. But if CTCF is not bound here, it can, so enhancers can control H19 as well as IGF2. Okay, so what happens is that, okay, so this region, this is present as it is in both maternal copy allele and the paternal allele. Okay, yeah. So this is CTCF binding site. Let us, okay. Pink one is CTCF binding site. Okay. So what happens that this cluster, this is paternally imprinted cluster. Okay. So means the DMR will be imprinted, or DMR will be that will be methylated in the paternal allele. So here DMR is the CTCF binding site. Okay, this is differentially methylated region. So this DMR, this will be methylated. So the shaded region means it is methylated only in the paternal allele. So this is the maternal copy and this is the paternal copy. Okay, so the DMR, this is paternally imprinted gene, the H19-IGF2 cluster. This is paternally imprinted, and that is why it is only methylated in the DMR. That will be methylated here. So what happens is that whenever CTCF binding site is methylated, so now CTCF can't bind to it. So in the paternal allele, as the binding site is methylated, CTCF cannot bind to here, but in the maternal allele, CTCF can bind because it is not methylated. So let's say that this is CTCF Okay, so now CTCF is bound here. That is why the enhancer can only control H19, but there will not be production of IGF2 because enhancer cannot now control the IGF2 gene, and that is why it will not be expressed. Why? Because now there is boundary element, there is CTCF here. But in the paternal allele, as the CTCF binding site is methylated, the enhancer can control both H19 as well as IGF2. So there will be expression of IGF2 as well as H19. Okay. So this is how the IGF2 inhibition in the maternal allele, this leads to growth suppression because IGF is the it is the growth factor, so it promotes growth. So there will be growth suppression. Okay, and in the paternal allele there will be growth promotion. Okay, so growth will be promoted. Why? Because the IGF2 is also expressed. So this is how the ICR, the DMR in the ICR, it regulates the genomic imprinting. So I hope everything that I discussed here was clear to you all. If you have any queries, you can ask them in the comment section below. In tomorrow's lecture, we will learn about certain syndromes which are linked to those clusters, those imprinted regions. If something goes wrong in this imprinted region, if there is some mutation, or if there is uniparental disomy, how that can affect and that can lead to syndromes or disorders. Till then, see you. Bye bye.